what if the exact same technology currently being hailed as a miraculous cutting-edge cure for terminal brain cancer is, also at the center of a growing global debate about actually causing it?
Yeah, it's a massive paradox to wrap your head around.
Right. Welcome to the deep dive, everyone. Whether you are, rigorously analyzing medical data for your profession, or you're just wildly curious about what exactly went into your arm a few years ago, this one is custom tailored for you.
It really is. And, when you look at the raw cellular biology, that paradox we just mentioned, it, it starts to make an uncomfortable amount of sense.
It does, and we have a formidable stack of literature to help us make sense of it today. I mean, we are looking at a 6-year retrospective on the COVID-19 mRNA rollouts.
Huge amount of data.
Yeah. We've got standard oncological guidance from breastcancer.org. We have systematic reviews from the journal OncoTarget. We're looking at theoretical frameworks published in Curious and preprints.org.
Not to mention the case reports.
Exactly. We even have a highly technical case report on bladder cancer from the International Journal of Innovative Research in Medical Science, alongside official CDC presentation slides, detailing safety uncertainties.
We are casting a very wide net today. I mean, we're pulling from mainstream clinical guidance that you'd hear in literally any doctor's office, all the way to highly technical, multi-omic case studies.
Right.
And systemic reviews that are just trying to process millions of data points over the last half-decade.
So here is the mission for today's deep dive. We are going to explore a very specific growing debate in the medical and legal literature.
Yes.
And the core of this debate argues that, by legal and medical definition, there's actually no COVID-19 vaccine 6 years later.
Which sounds crazy, but...
Right. The argument is that their unique mechanisms, specifically the delivery via lipid nanoparticles and the ribosomal synthesis of the spike protein, fundamentally classify them as prophylactic immunogene therapies, or mRNA therapeutics, rather than traditional vaccines.
And, it's hard to look at this data without all the political and social noise of the last 6 years creeping in.
Oh, absolutely.
But looking at this stack of literature, we really just need to strip all that away. We need to look strictly at the cellular biology.
Just the facts.
Right. Our job today is purely to impartially report, synthesize, and unpack the factual content contained in these specific source documents.
Yeah.
Just looking at the mechanisms exactly as they are written in the clinical and scientific literature.
That is really the only way to do it. So, to understand the safety signals and the oncology discussions we're going to get into later, we first have to fundamentally redefine what these mRNA shots actually are.
Yeah, that's the baseline.
Why are researchers in the preprint source arguing so heavily against the word vaccine?
Well, the terminology matters deeply because the mechanism is completely unprecedented in the history of global immunization. To understand why scientists are pushing for the term immunogene therapy, we have to look at what the medical and legal definition of a vaccine used to be. Historically, a vaccine works by introducing an antigen.
Right. Like a piece of the actual virus or bacteria?
Exactly, into the body to provoke an immune response.
Right, like the Novavax COVID-19 shot or, your standard childhood DTaP shot.
Precisely. In those traditional platforms, pharmaceutical companies manufacture the antigen in a laboratory.
Outside the body.
Right. They use massive bioreactors, they purify the viral protein, put it in a vial, and inject that piece of the invader directly into your arm.
Got it.
So the factory is entirely outside the human body. Your immune system sees the foreign debris, mounts a defense, and creates memory B cells and T cells to remember it.
But with Pfizer's Comirnaty and Moderna's Spikevax, there is no protein in the vial.
None. Zero. They inject genetic code.
Yeah.
Specifically synthetic messenger RNA. They are injecting a biological blueprint. And because of this, the preprints.org authors argue that the mRNA platform fundamentally transforms the entire process of antigen processing and presentation.
How so?
Well, you are not receiving an antigen. You are receiving the genetic instructions to become the factory that makes the antigen.
Okay, let's unpack this. How does changing the location of the factory, like from a lab in New Jersey to the cells in my arm, actually transform the biological process?
It's a great question.
Because isn't the destination the same? The immune system eventually sees the spike protein either way, right?
Well, the destination seems the same on the surface, but the biological pathway to get there is entirely distinct. And that distinction carries profound risks.
Okay.
It comes down to something called ribosomal synthesis.
Yeah.
In a traditional vaccine, the antigen is floating in the extracellular space, which is the fluid outside your cells.
Right.
Specialized immune cells, called antigen-presenting cells, swallow it up, chop it to pieces, and display the pieces on their surface. This is a normal external threat assessment.
Like finding a piece of trash in the yard and identifying it.
Yes, exactly. But with mRNA, the dynamic flips entirely.
Oh.
The mRNA is absorbed directly into the host cells. Once inside, it escapes into the cytoplasm and hijacks the host cell's ribosomes.
The ribosomes, those are like the 3D printers of the cell, right? They read genetic code and print proteins.
That's a great visualization, yeah. The ribosomes, whether they are free-floating in the fluid of the cell or attached to a structure called the rough endoplasmic reticulum,
Okay.
they read the injected mRNA blueprint and begin manufacturing the SARS-CoV-2 spike protein internally. This is called in situ synthesis. Synthesis happening inside your own tissue.
And why is that inherently a problem?
Because when a host cell manufactures a viral protein internally, it processes that protein differently. It often expresses the newly minted spike protein directly on its own plasma membrane.
Oh.
Yeah. Now, instead of the immune system just seeing a piece of floating debris in the extracellular space, your cytotoxic T cells.
The assassins of the immune system.
Right. They see your own cells expressing a viral protein.
Oh. Wait, so the T cells don't know that my cell was just handed a blueprint?
Yep.
They just see a human cell covered in viral spikes.
Exactly. And they assume that cell is infected by a live virus. This triggers a process called apoptosis, or programmed cell death.
Wow.
Your immune system is actively destroying your own tissue. It is the difference between showing the police a picture of a burglar versus dressing your own citizens up as the burglar.
Oh, man.
The police are going to attack the citizens.
That is a terrifyingly clear analogy. If we are dressing our own cells up as the burglar, it immediately begs the question of which cells are getting dressed up.
Exactly.
Which brings us to the delivery vehicle, because if the mRNA is just the blueprint, how does it actually get inside the cellular factory?
Right.
I always thought natural RNA degrades almost instantly if you just inject it.
It does. The body is full of enzymes called ribonucleases that are specifically designed to chop up free-floating rogue RNA on contact.
As a defense?
Yeah, it's a defense mechanism against RNA viruses. So to survive that gauntlet, the synthetic mRNA requires a highly specialized delivery vehicle. We're talking about lipid nanoparticles, or LNPs.
Right. The early public perception was that these were just microscopic bubbles of fat. Just inert, harmless packaging to keep the mRNA safe until it got to the cell.
The preprints and Curious papers make it abundantly clear that this perception was entirely inaccurate.
Not just fat bubbles.
Not at all. These are not inert bubbles. They are pharmacologically active, highly inflammatory immune stimulants.
Oh.
The sources detail the specific structures used. Pfizer uses a proprietary blend of lipids named ALC0315 and ALC0159. Moderna uses SM102 and DMG.
Okay, I noted something really interesting in the sources about these specific lipids.
Yeah.
These ionizable lipids were originally designed for gene therapy. There's a drug called patisiran, or Onpattro, which is used for a rare liver condition called amyloidosis.
Yes.
And these lipids were chosen specifically because they are fusogenic. What does that actually mean?
Fusogenic means they're explicitly designed to actively fuse with human cell membranes. An ionizable lipid is a marvel of bioengineering, really. It actually changes its electrical charge based on the acidity of its environment.
How does that help it get into the cell, though?
Well, when the LNP approaches a cell, the cell often swallows it into a tiny internal pocket called an endosome.
Okay.
The cell is trying to figure out what this thing is. Now, the environment inside that endosome is highly acidic.
Right.
When the ionizable lipid hits that acid, it takes on a positive charge. This shift in charge causes a violent physical reaction.
Really?
Yeah, it literally ruptures the endosome and dumps the mRNA payload directly into the cell's cytoplasm.
So it's not a gentle delivery. It's aggressively breaking into the cell's interior.
It is highly aggressive.
Yeah.
Which is why it's so effective as a gene therapy tool.
Yeah.
But this aggressive penetration is also highly inflammatory to the cell.
Okay, so if these LNPs are that good at penetrating cells, where do they go once they enter the body?
That's the million-dollar question.
Because I remember early on, the standard line, the one we heard everywhere, was that the injection stays right there in the deltoid muscle of your arm. It stays local.
The CDC slides and the Curious data we reviewed paint a completely different picture regarding biodistribution.
Really?
Yeah. The preclinical rat and mouse studies that the company submitted to regulators, along with subsequent human data, show rapid and extensive multi-organ distribution.
It doesn't stay in the arm.
Not at all. Within hours, High levels of the LNP mRNA complexes are found in the draining lymph nodes, the liver, and the spleen.
Wow.
They also distribute, in varying concentrations, to the ovaries, the testes, the adrenal glands, and the eye.
The eye.
Yes, trace amounts are found in ocular tissue. And perhaps most critically from a neurological standpoint, the pharmacokinetic data shows that a small fraction, roughly 2 to 4% of the plasma levels, actually crosses the blood-brain barrier.
Wait, hold on. The blood-brain barrier is supposed to be the ultimate fortress. It keeps toxins and pathogens out of the brain. You're saying these LNPs cross it.
Because of their microscopic size and lipid composition, yes, a percentage can cross the barrier.
That's wild.
This means we are talking about a systemic distribution of a fusogenic particle that forcefully enters cells across multiple organ systems, instructing whatever cell it enters to manufacture a foreign spike protein.
So we have citizens dressed up as burglars in the liver, the spleen, the ovaries, the heart, and potentially the brain.
Exactly.
How long does this blueprint stick around? If the body is turning into a factory, how long does the factory stay open?
Wow.
Because the CDC slides highlight a detail about persistence that I think a lot of people would find shocking.
Oh, absolutely. The official CDC slides explicitly note that vaccine mRNA persists in the axillary lymph nodes, which are the nodes under your arm,
Right.
and in the heart tissue, specifically the myocardium, for up to 30 days.
30 days.
And it is crucial to note that 30 days was simply the endpoint of the specific biopsies they were referencing.
Oh, I see.
It didn't disappear at day 30. They just stopped looking.
A month. The factory's open for at least a month, churning out these proteins.
And before we even talk about the damage those proteins might do,
Hmm.
there is another layer to the LNP structure itself that we have to consider, which is the PEG coating, polyethylene glycol.
I've seen that term. Why is it coated in PEG?
The LNP is incredibly unstable on its own. It's coated in PEG to stabilize it and keep the billions of nanoparticles from clumping together in the vial.
Okay, that makes sense.
But PEG is inherently immunogenic, meaning the human body can develop antibodies against it. In fact, a large percentage of the population already has anti-PEG antibodies from exposure to everyday cosmetics and laxatives.
If we connect this to the breastcancer.org source, this is highly relevant in oncology.
Yes.
That source notes that cancer patients are explicitly warned to speak with their doctors about PEG and a related compound, polysorbate.
Right.
Why? Because some patients have severe, life-threatening allergic reactions to pegylated cancer drugs like Doxil or Neulasta.
It is a vital connection. We are using a highly inflammatory systemic delivery vehicle coated in a synthetic compound known to trigger severe hypersensitivity and anaphylaxis in a subset of the population.
Wow.
And that vehicle is delivering a highly engineered synthetic payload.
Let's talk about that synthetic payload, because we've delivered the blueprint to the organs, but as you mentioned earlier, natural RNA is incredibly fragile.
Very fragile.
Even if it gets inside the cell has defenses. So how did the vaccine makers force the human body to keep reading this blueprint for 30 days or more without destroying it?
They achieved that by fundamentally altering the chemical structure of the RNA itself. This specific modification is actually what recently won the Nobel Prize in medicine.
Oh, right.
Yeah. In natural messenger RNA, you have 4 basic building blocks called nucleosides: adenine, cytosine, guanine, and uridine.
The letters of the genetic code.
Exactly, the A, C, G, and U. The researchers discovered that if they replaced every single natural uridine molecule in the entire sequence with a synthetic lab-created analog called N1-methylpseudouridine.
That's a mouthful.
It is. But when they did that, the mRNA suddenly became incredibly rigid and stable. It resisted being degraded by the cell's internal nucleuses.
But the curious paper explains that the pseudouridine doesn't just act as armor. It actively deceives the cell.
It does. The human innate immune system has an ancient, highly evolved security system. It uses sensor proteins called toll-like receptors, specifically TLR3, TLR7, and TLR8.
Okay.
These receptors sit inside the cell's endosomes and cytoplasm, acting like biological bouncers. Their entire job is to detect foreign invading RNA from viruses and sound the alarm by triggering massive inflammation and shutting down the cell's operations.
Wait, so it's basically a chemical fake ID? The bouncers don't recognize it?
Fake ID is the perfect way to synthesize it. The bouncers, the toll-like receptors, look at the synthetic pseudouridine, and because it has a slightly different molecular shape than natural viral uridine, they don't recognize it as a threat.
That's wild.
Yeah, the synthetic mRNA slips past TLR3, 7, and 8 completely undetected.
So the alarm never sounds?
The alarm is silenced. This is what allows the synthetic mRNA to survive long enough to produce massive amounts
Hmm.
of the spike protein without the cell immediately destroying itself from the inside out to stop the viral replication.
I see.
In clinical terms, the pseudoiodine ablates or heavily dampens the type I interferon response.
Type I interferon, that's the chemical signal the cell sends out to warn the rest of the immune system.
Yes, it is the primary antiviral alarm bell.
But there has to be a cost to that evasion. If you are artificially dampening the toll-like receptors and the type I interferon response just to sneak your vaccine payload in, what happens to the rest of the body's security system?
This is where the theoretical risks become dangerously systemic.
Yeah.
And this is the core focus of the curious hypothesis.
Okay.
Evading the TLRs prevents the patient from going into immediate fatal anaphylactic shock upon injection, which is why the Nobel Prize was awarded. It solved the immediate toxicity problem of RNA therapy.
Right.
But by dampening the type I interferon response, you might create a transient window of severe immunosuppression.
Meaning the immune system's guard is temporarily lowered across the board. The bouncers are asleep.
Yes. And a robust, active TLR-mediated interferon response is absolutely necessary to keep other biological threats in check. We all harbor dormant viruses.
Like what?
Most adults carry varicella zoster, which causes shingles, or the Epstein-Barr virus. And crucially, our bodies are constantly policing microscopic, latent cancer cells.
Wait, so our immune system is fighting off cancer all the time?
Constantly. It's called immunosurveillance. Key cells and natural killer cells are always patrolling, looking for cells that are mutating and eliminating them before they can form a tumor.
Wow, I had no idea.
Yeah. And the curious authors raise a very stark concern. By chemically forcing the bouncers to look the other way for days or weeks, we might be creating an immunological blind spot.
Oh, no.
A blind spot wide enough to allow dormant viruses to reactivate, which we saw with the massive spike in shingles post-vaccination. Or worse, allowing latent cancer cells to escape surveillance and establish a foothold.
That is deeply concerning. And as if deliberately suppressing the immune system wasn't complicated enough, we also have to talk about how the ribosomes actually read this fake ID.
Right, the translation process.
Because the CDC slides bring up a phenomenon called frame shifting. What is that?
Frame shifting is a fascinating and somewhat unpredictable consequence of using heavily modified, hyperstabilized synthetic RNA. To understand it, you have to know how translation works.
Okay.
The genetic code is read by the ribosomes in sets of 3 letters at a time, called codons.
Like reading 3-letter words, C-A-T-D-O-G.
Exactly. But because the pseudoiodine is synthetic and physically bulkier than natural uridine, and because the mRNA is engineered to translate at maximum possible speed, the ribosomes can sometimes slip as they read the sequence.
Slip. Yeah.
They lose their place by 1 or 2 letters.
Like skipping a groove on a vinyl record. So instead of reading C-A-T, it shifts over 1 letter and reads A-T-D.
Precisely. And when the ribosome slips, the entire downstream sequence is now read out of order. Instead of producing the intended SARS-CoV-2 spike protein, the cell begins manufacturing entirely random, off-target nonsense proteins.
Wait, really?
Yes. The CDC slides note this phenomenon, and a recent landmark paper confirmed that these off-target proteins are indeed produced in vaccinated individuals, and the human immune system actually mounts a response to them.
Wait, our cells are manufacturing random, unpredictable proteins, and our immune system is attacking them. What does that mean for the patient?
The honest scientific answer right now is that the long-term effects of these off-target, frame-shifted proteins are entirely unknown.
That's unsettling.
It is. We don't know what they do. We don't know if they form aggregates like the amyloid plaques in Alzheimer's. We don't know where they accumulate or what kind of chronic autoimmune conditions they might trigger over a decade.
Okay, let's just pause and summarize the sheer biological chaos we've uncovered so far.
It's a lot.
We've got a fusogenic lipid nanoparticle that breaks into cells systemically, liver, heart, brain. It's carrying a pseudoiodine fake ID that suppresses the innate immune alarm system.
Right.
It's occasionally stuttering to produce random nonsense proteins. And it's doing all of this while forcing the body to act as a factory for the main product, the SARS-CoV-2 spike protein.
Yes.
Let's look at that main product, because what happens when the factory produces too much or packages it uniquely?
Well, we have to address the inherent toxicity of the spike protein itself.
Okay.
For a long time, The public health messaging treated the spike protein as just a neutral structural component of the virus,
Yeah.
like a piece of Velcro the virus uses to attach to cells.
Yeah, that's what I remember hearing.
But the preprint source synthesizes a wealth of literature demonstrating that the spike protein is highly toxic and pathogenic in its own right, even without the rest of the virus attached to it.
Toxic how?
It acts almost like a bacterial superantigen or a toxoid.
A superantigen, like the toxins that cause toxic shock syndrome from a bacterial infection?
Very similar, yeah, in its capacity to provoke an out-of-control hyperinflammatory immune response. The literature indicates that the spike protein causes severe endothelial inflammation.
Endothelial meaning the lining of the blood vessel.
Yes, it damages the smooth lining of your veins and arteries. It disrupts the blood-brain barrier. And crucially, it interacts directly with platelets and coagulation pathways to induce microvascular clotting.
Oh, wow.
And remember the biodistribution we discussed?
Mm-hmm.
Because of the LNPs, this isn't just happening in the respiratory tract like it would in a natural, mild respiratory COVID infection.
Right, it's everywhere.
It's happening systemically, deep inside whatever organs the LNPs landed in.
And it gets even stranger because of how the host cells package the spike protein once they manufacture it.
Yeah, the exosomes.
Right. The sources detail a cellular behavior involving exosomes. Can you explain what these are?
Exosomes are essentially tiny, natural lipid vesicles, little biological envelopes that cells use to communicate with each other. They pinch off from the outer cell membrane, carrying proteins and genetic material through the blood to distant parts of the body to send signals.
Like cellular mail.
Yes. What researchers are finding is that human cells transfected by the vaccine are packaging the newly manufactured spike protein along with intact fragments of the synthetic mRNA into these exosomes and releasing them into the bloodstream.
So they aren't just expressing the spike on their surface for the immune system to find, they are mailing it out to other organs.
Exactly. And what's fascinating and deeply concerning is that these exosomes act like dumbed-down virus LNP chimeras.
Chimeras.
Yeah. These circulate through the lymphatic and vascular systems. They essentially spread the spike protein to distant tissues, mimicking the behavior of a real disseminating virus.
Wow.
They act as mobile antigen-presenting units, traveling far beyond the initial site of transfection. This provides a mechanism for continuous systemic antigen dissemination, potentially triggering inflammatory immune responses in distant organs weeks or even months after the injection.
Which perfectly sets up the most heated high-stakes debate in all the literature we reviewed today, the intersection of this mRNA technology with oncology.
It's a heavy topic.
We know this system distributes inflammatory LNPs, it circulates spike-laden exosomes, and it temporarily suppresses innate immunity via the pseudoridine. So how is all of this impacting cancer rates?
This is where we see a stark, almost jarring divergence between mainstream clinical guidance and the emerging retrospective literature. Let's look at the standard guidance first so we understand the baseline.
Right. The breastcancer.org source anchors us in what you will hear in almost any standard clinical setting today.
Yes.
Mainstream medical guidance actively recommends the 2025-2026 COVID-19 shots for cancer patients. They explicitly state there is no evidence linking the vaccines to cancer causation or recurrence.
Correct. That's the current establishment stance.
The main clinical issue they note is purely mechanical. They warn that the LNPs cause intensely swollen axillary lymph nodes under the arm, which can mimic a breast lump and cause false positive mammograms. They even advise patients to get the shot in the opposite arm of their breast cancer to avoid confusing the radiologist. It's framed as an imaging nuisance, not an oncological threat.
That is the established institutional consensus. But when we look at the systematic reviews, like the comprehensive one published in OncoTarget by researchers Cooper Wasser and L. Derry,
Right.
a completely different, highly concerning signal emerges. This review isn't just one doctor's opinion. It looked at 69 peer-reviewed publications, covering 333 patients across 27 countries.
That's a lot of data.
And they layered that with massive population-level demographic data from Italy, Korea, and the U.S. military databases.
And what are the researchers in the OncoTarget study actually seeing in these patients?
They are documenting recurrent themes that completely defy standard oncological timelines.
How so?
Well, in oncology, tumors generally follow predictable growth curves. But these researchers are seeing unusually rapid progression, sudden recurrence, or violent reactivation of pre-existing indolent cancers.
Indolent meaning slow-growing or previously well-controlled by treatment.
Exactly. Cancers that were in remission for years suddenly exploding. They're also noting atypical histopathological findings,
What does that mean?
meaning when they look at the tumor tissue under a microscope, it looks bizarre. They're seeing aggressive tumors erupting right in the vaccine injection sites on the arm, or heavily concentrated in the draining regional lymph nodes where we know the LMPs accumulate.
And are they seeing specific types of cancer? Or is it just across the board?
There is a marked disproportionate uptick in hematologic malignancies, so blood cancers,
Okay.
things like non-Hodgkin's lymphoma, Hodgkin's lymphoma, and cutaneous lymphomas. They're also seeing aggressive solid tumors like sarcomas and melanomas. The U.S. military data they cite specifically mirrors this massive rise in lymphomas during the peak mandate period.
So we have a total contradiction. Mainstream guidance says it's perfectly safe, but researchers in OncoTarget and Curious are documenting explosive tumor growth.
Yes.
How do these researchers explain these sudden cancer signals? What is the actual biological mechanism they are proposing that links the shot to the tumor?
They are proposing a framework called the multi-hit hypothesis of oncogenesis.
Multi-hit.
Yeah. It suggests the vaccine doesn't necessarily enter a perfectly healthy cell and magically turn it into a cancer cell from scratch.
Okay.
Rather, it creates a perfect storm of environmental conditions for existing microscopic, dormant cancer cells to escape and explode into hyperprogression.
How does that storm form? Walk me through the steps.
It is a combination of the factors we've been unpacking. Hit number one, you have the intense, massive immunostimulation from the inflammatory lipid nanoparticles.
Right.
This causes a systemic cytokine storm, a flood of inflammatory signals. Cancer thrives on inflammation. It uses it like fertilizer.
Okay, so fertilizer, what's hit number 2?
Hit number two is the immune evasion tactics of the pseudocytine. Remember the fake ID?
The bouncer's looking the other way.
Exactly. It dampens the type I interferon response and drastically lowers the activity of CD8 plus cytotoxic T cells. Those are the very cells responsible for hunting down rogue cancer cells. So you've just knocked out the immune system snipers.
Wow. And hit number 3.
The persistent spike protein itself. The spike protein drives chronic inflammation and has been shown to physically alter cellular signaling pathways, specifically the MAPK and PI3K pathways.
I saw those acronyms, MAPK and PI3K. What do they actually do?
Think of them as the cellular gas pedals for cell division and growth.
Oh.
When those pathways are activated, the cell is told to multiply. The spike protein essentially presses down on the cellular gas pedal.
Oh, wow. So if we synthesize this, you have massive inflammation feeding the tumor environment. You have the spike protein pressing the gas pedal for cell growth,
Yep.
while simultaneously the pseudocytine knocks out the immune system's snipers that would normally stop the growth.
Precisely. This profound multi-pronged imbalance overrides the body's natural immunosurveillance. Dormant cancer cells suddenly find themselves in an environment where the guards are asleep, the gas pedal is stuck to the floor, and growth-promoting inflammatory signals are everywhere.
That's a nightmare scenario.
The result is immune escape and tumor hyperprogression.
Okay, that is a compelling, terrifying theory. But let's go even deeper. Let's look at the factory floor itself.
Oh, the contaminants.
Because beyond the mRNA, beyond the LNPs, and beyond the spike protein, the manufacturing process itself left something else behind in those vials. Something that brings us to the most controversial, highly technical, frankly mind-bending part of our deep dive today.
You are referring to the plasmid DNA impurities.
Yes. The official CDC slides and the preprint source reveal that Pfizer and Moderna vials contained residual, fragmented plasmid DNA from the manufacturing process.
Right.
And not just trace, negligible amounts.
Yeah.
Depending on the batch, they exceeded regulatory safety limits by 36 to 627-fold.
It's astonishing.
And crucially, the researchers found that the Pfizer vials contained a specific genetic sequence called the SV40 promoter enhancer.
This is perhaps the most critical detail in the entire genomic debate. We have to understand how these shots were manufactured at scale.
How are they?
You can't just synthesize billions of doses of mRNA in a test tube using chemicals.
Yeah.
The companies used E. coli bacteria as biological vats.
Brocaterial.
Yeah. They engineered massive quantities of circular DNA called plasmids, which contain the master blueprint for the spike protein, and put them inside the bacteria to multiply.
They grew the blueprints in bacteria.
Yes. Then they harvested the bacteria, burst them open, extracted the DNA plasmids, chopped them up, and transcribed that DNA into the mRNA for the vaccine.
Okay.
But the purification process, the step meant to filter out all the leftover DNA and bacterial sludge, it was imperfect. Billions of fragments of this chopped up plasmid DNA were left behind in the final vials.
Wow.
And because they were in the same vat, They were packaged right alongside the mRNA inside those fusogenic, cell-penetrating LNPs.
So the LNP isn't just delivering the mRNA blueprint, it's delivering fragmented DNA.
Exactly.
And what is the SV40 promoter sequence that was found in the Pfizer vials? Why is that significant?
The SV40 promoter is a genetic sequence originally derived from the simian virus 40. In molecular biology, an SV40 promoter is famous. It is an incredibly powerful tool used in labs to drive massive gene expression.
Oh, okay.
But more importantly, it contains a nuclear localization signal. It is a known biological vector specifically designed to push DNA straight into the nucleus of host cells and facilitate integrating it directly into the human genome.
What?
It acts as a microscopic Trojan horse.
It pushes the DNA into the nucleus, where our human chromosomes are.
Yes.
Which brings us to the most startling document in our stack, the clinical case study published in the International Journal of Innovative Research in Medical Science, the bladder cancer case.
Yes. This case study is a sentinel event in the current literature.
Yeah.
It meticulously documents the clinical timeline of a 31-year-old, previously perfectly healthy female.
Okay.
Within 12 months of completing a 3-dose Moderna mRNA series, she developed rapidly progressive stage IV bladder cancer.
Wait, bladder cancer in a 31-year-old female? That has to be incredibly rare on its own, isn't it?
Exceptionally rare. Bladder cancer is typically a disease of elderly men with a long history of smoking or chemical exposure.
Right.
An advanced stage IV presentation with distant metastases is almost unheard of in a healthy 31-year-old woman. Because of this extreme anomaly, the treating researchers decided to look deeper.
What did they do?
They performed comprehensive multi-omic profiling on her tumor tissues, her blood, and her urine.
I see the term multi-omic profiling, utilizing tools like PBME and Revis. What does that actually mean? What are they looking at?
Imagine if you were investigating a factory that was producing defective cars. Standard testing might just look at the final car. Multi-omic profiling looks at everything.
Okay.
It sequences the entire genome, which is the DNA blueprint, the transcriptome, the RNA messages being sent, the proteome, the actual proteins being built, and the metabolome, the waste products.
That's incredibly thorough.
PBMA and Revis are just highly advanced computational algorithms used to scan all this data and find microscopic anomalies, like looking for a single typo in a million-page encyclopedia.
And when they scanned her entire biological factor, they found total chaos.
They found profound molecular dysregulation. The algorithms identified hyperactive oncogenic driver genes. These are the specific genes that, when mutated, cause cancer to grow out of control.
Like which ones?
Genes like KRAS, ATM, and MPK1 were wildly dysregulated. They also found severe DNA repair deficiencies, specifically in a crucial gene called MSH2.
So the genes that drive cancer were stuck in the on position, and the genes that fix broken DNA were broken themselves. The genome was essentially fracturing and unable to fix itself.
Exactly. But the genomic chaos alone doesn't prove a link to the vaccine.
Right. But then they found a smoking gun, didn't they? They found a host vector chimeric red. Explain what that is in simple terms.
A chimeric read in DNA sequencing means the computer found a piece of human DNA physically fused end-to-end to a piece of foreign artificial DNA.
Wow.
Specifically, they found a 20 base pair fragment of the vaccine plasmid DNA physically and permanently integrated into the patient's human chromosome 19.
Wait, it actually integrated into her genome? The vaccine DNA became part of her human DNA?
Yes, it integrated.
Where exactly did it land?
It landed at a specific location called cytoband 19q13.42. To understand why this specific location matters, you have to know about the concept of the AAVS1 safe harbor.
Safe harbor.
In the human genome, there are designated safe harbors. Regions of junk DNA where if a foreign piece of DNA happens to insert itself during gene therapy, it probably won't disrupt any vital genes or cause cancer. It just sits there silently.
But this fragment didn't land in a safe harbor.
No, it landed just outside it. It integrated in a highly transcriptionally active, recombination-prone regulatory region, about 158 kilobases upstream of something called the ZNF gene cluster.
What does that mean for the cell?
It means it landed in a densely packed, highly active neighborhood of the genome. Inserting a foreign piece of DNA here carries a massive risk of transcriptional disruption.
Like breaking the machinery.
Yeah, it can warp the folding of the chromosome, create mutated fusion proteins, and drive the exact kind of severe genomic instability we saw with her MSH2 and KRAS genes. It is like throwing a wrench into a highly complex set of moving gears.
But wait, I noticed a massive paradox in the source data when I was reading it.
Oh, the Moderna versus Pfizer thing?
Yes. The patient received 3 doses of the Moderna vaccine, but the paper states the integrated DNA was a perfect 20 for 20 base pair match to the Pfizer BNT162b2 plasmid. How is that even possible? Did she get the wrong shot?
It's a great question, and it highlights a major issue with corporate transparency and shared manufacturing architecture.
What do you mean?
The researchers used a global genomic database called BLAST to identify the foreign DNA fragment. The sequence they found matched a specific segment within the spike open reading frame, which is the actual code for the spike protein itself.
Right.
The reason it matched Pfizer in the computer system is simply because Moderna has never published or deposited their proprietary plasmid sequence into the public NCBI database.
Oh, so the computer just found the closest known match available to the public.
Exactly. The genetic code for the spike protein is virtually identical across both the Pfizer and Moderna platforms. Because Pfizer's plasmid code was leaked and made public, the database defaulted to Pfizer.
That makes sense.
It proves cross-platform structural homology. The core blueprint is the same. And we must emphasize the statistical weight of this finding. The mathematical probability of a random 20-base sequence perfectly matching a predefined target in the human genome is roughly 1 in a trillion.
Wow.
It is statistically impossible for this chimeric red to be a random computer artifact or a sequencing error. It is a physical reality.
So the DNA integration actually happened. It physically altered her chromosome. What are the broader implications of this?
Well, while we must impartially and rigorously state that a single case report does not definitively prove causation for all cancers across the global population, the implications of this single finding are staggering. It proves that integration of vaccine-derived DNA into human chromosomes is biologically possible in vivo, inside a living human being.
It's not just a theory in a petri dish anymore.
No, it opens the door to the reality of insertional mutagenesis, the process where foreign DNA breaks a human gene and directly causes cancer. And it provides a highly plausible, documented molecular mechanism for the controversial turbo-cancer hypothesis observed in the massive oncotarget dataset.
Okay, we've spent almost this entire deep dive looking at how this technology, the LNPs, the pseudoridine, the frame shifting, the DNA integration, how it might accidentally provoke or accelerate cancer.
Yeah.
It sounds like an absolute nightmare. But to fully understand immunogene therapy, we have to look at the flip side of the coin. There is a duality to this technology that is just as mind-bending.
There is, and it is a profound, almost poetic, double-edged sword of modern medicine.
Let's bring in the final source from UF Health. Scientists like Dr. Elias Sayer and researchers at Johns Hopkins are using the exact same technology, lipid nanoparticles and modified mRNA, to fight cancer.
Yes, they are.
They are seeing incredible, unprecedented results in human trials using mRNA to cure highly lethal, previously untreatable tumors like glioblastoma.
And if we synthesize this duality, it makes perfect, ruthless biological sense. The very mechanisms that make the COVID-19 vaccines potentially risky and chaotic as prophylactic shots in healthy people are the exact same mechanisms oncologists are desperately trying to harness to save dying patients.
Walk me through that. How does the same weapon heal and harm?
Think about a highly lethal, aggressive tumor like a glioblastoma in the brain. It survives by hiding from the immune system. It creates an immunosuppressive microenvironment. It essentially puts the local immune cells to sleep so it can grow undetected. What the UF Health researchers are doing is taking an mRNA blueprint of the patient's specific tumor, putting it inside one of these highly inflammatory LNPs, and injecting it directly into the patient.
They are purposefully using the inflammatory vehicle.
Yes. They are purposefully utilizing the massive localized inflammation, the aggressive forced cellular production of foreign proteins, and the extreme violent activation of the immune system.
Wow.
They want to trigger a cytokine storm inside the tumor. They are using the toxicity to wake up the patient's dormant immune defenses and point them like a laser at the tumor cells.
They are weaponizing the chaos. In terminal cancer, massive immune activation and cellular destruction are exactly what you want. You want to destroy the tissue.
Exactly. You are trying to detonate a bomb inside a tumor. But when you take that exact same mechanism, that same bomb, and apply it as a preventative, widespread prophylactic measure in billions of perfectly healthy individuals, you are introducing a highly disruptive inflammatory weapon into a peaceful environment. You are injecting chaos where there was order.
Wow. Okay. We have covered a massive amount of incredibly dense terrain today.
We really have.
From fundamentally redefining these shots not as traditional vaccines, but as prophylactic immunogene therapies, We've explored the aggressive, fusogenic properties of lipid nanoparticles, the fake ID evasion tactics of synthetic pseudoridine.
The systemic spread of spike-laden exosome chimeras.
Yes, and the intense high-stakes debates surrounding cancer safety signals and actual DNA integration into human chromosomes.
It is a staggering amount of highly technical biology to process. But, whether you've received 0 doses or 6, understanding this underlying technology is absolutely vital for your future health literacy.
Why is that?
Because mRNA platforms are not going away. They are the new frontier. They're currently in advanced clinical trials for influenza, RSV, HIV, and, as we just discussed, personalized oncology.
Right.
This is undeniably the future of medicine. But the safety surveillance parameters, the regulatory oversight, and our understanding of genomic integrity must evolve rapidly to match the sheer unfiltered power of this technology.
It has to, because if it doesn't, we are flying blind into the next decade of medicine. And I want to leave you, the listener, with a final thought to mull over today as you process all of this.
Go for it.
We talked about how, with mRNA immunogene therapy, your own body becomes the factory. I want you to think about the logistics of that. In traditional medicine, a pharmaceutical factory in a controlled lab makes a pill with a precise, measured dose of a drug.
Right.
You swallow it, and your doctor knows exactly how many milligrams are in your bloodstream.
It is predictable.
Exactly. But when your body is the factory, every human being is completely different. A 110-pound woman has a wildly different metabolic rate than a 250-pound male athlete.
Absolutely.
Someone with an underlying autoimmune disease processes cellular stress differently than someone who is perfectly healthy. If the LNPs distribute systemically and the ribosomes churn out proteins until the RNA degrades, how does the manufacturer control the dose?
They don't.
They can't. And that is the ultimate paradox of this technology. We have engineered the most advanced biological delivery system in human history, but we have surrendered control of the final dose to the unpredictable chaos of the human body. Thank you for taking the deep dive with us.